Hello everyone, we complete our lesson today for grade 11 advanced, and today we're going to talk about electron transport. Now just to refresh and go over the things that we discussed in previous lessons, we'll look over the overview of the light-dependent reaction, so that is the electron transport. We're going to look at what are the things that we have in this process, and we're going to go over the steps of this process. Before, I just want to let you know that I'm going to first start by explaining the electron transport through the diagrams provided in the book, through the explanation provided in the book, and then I will be going over a summary of this whole process using the images and videos provided from Aleph. Since the book diagram isn't very neat or very descriptive, the ones on Aleph are a bit more simple and a lot easier to understand. So this video is going to be containing the explanation from the book, and then in a separate video, I'll add the explanation using the Aleph diagrams. Okay, but it's good to note that explanation will only be a summary, just a quick overview of the process again. I won't be explaining in as much detail as I am now from the book, just for everyone to know. Okay, so let's begin. Let's go over an overview of the light-dependent reaction. So we have the light-dependent reaction, also known as the electron transport chain. Okay, so where is the site of this reaction? Where does it happen? It happens in the thylakoid. And if anyone remembers from our last video, the previous part of this lesson, we know that the thylakoids are found in the chloroplast, which is an organelle of the plant cell. What does the process need? Or what does the process do? You can say it needs light energy. The process needs light energy to take place. And we will see in a moment why we need the light energy as we look at the specific steps. It is important to note that water is essential for this process. So water is essential. It is very important for this process. So water is essential for electron transport. What are the products of this reaction? The products are, you have the energy store molecule ATP, and you have the reduced electron carrier NADPH. And I'll just explain why we say reduced. So when we say reduced, I'll just write it down here. So reduced electron carrier. We'll talk about electron carriers more in detail in a minute when we discuss the NADPH, but I just want to discuss reduced. Why did we say reduced electron carrier? When we say reduced electron carrier, it basically means, or when I say reduced, it means a molecule that accepted electrons. And this will make more sense in a moment when we discuss the steps. So we have ATP, the reduced electron carrier NADPH. NADPH. And we have oxygen released as a byproduct. So O2 as a byproduct. What do we mean when we say byproduct? We mean it is not something that we needed. So we did not do the reaction because we needed oxygen gas, but as a process of the reaction, we got oxygen gas as a product. I don't need it. It's a byproduct, and we'll see how it gets released as we look into the steps. Now, let's look at some of the components that we are going to be working with. If anyone wants to just write this down, I'll give you a second. So now we're going to look at the components of the thylakoid membrane. And I'd like to discuss the components first and look at, we'll learn more a bit about them before we go into the 5 steps of this process, or the 5 main steps of this process. So let us discuss the components of the thylakoid membrane. Okay, so the thylakoid membrane is the site of the light-dependent reaction of photosynthesis, as we have discussed. Now, we're gonna encounter something called photosystems. Photosystems. And photosystems are large clusters of proteins. So they're large clusters, and clusters basically means groupings. So large groupings, aka clusters, large groupings of protein and pigments. And when we say pigments, we know that we mean the light-absorbing molecules that exist within the thylakoid membrane. Now, these are functional units. So they are functional units for photosynthesis. So they're functional units for photosynthesis. And we have 2 types of photosystems, and they are photosystem 1 and photosystem 2. It is more accurately that I say we have photosystem 2 and then photosystem 1. You might be wondering why photosystem 2 comes first. It is because photosystem 1 was found first, but photosystem 2 is actually the one that starts the process. So if anyone's wondering why is it 2 then 1, it's because 1 was discovered before 2, but in actuality in the process, we use photosystem 2 and then we use photosystem 1. If anyone would like to highlight the information that we just discussed in the book, it is found right here. Oh, it is important to note, just before I show you where that is, that the thylakoid membranes have a large surface area. And this is important. Why? Because it provides the space needed to hold large numbers of electron transporting molecules and 2 types of protein complexes called photosystems. So it is important to note that the thylakoid membrane has a large surface area. And why do we need a large surface area? Because we need to hold large members of electron transporting molecules. I mean, it's in the name, electron transport chain. So we're moving electrons through a chain. So we need electron transporting molecules. And we have 2 types of protein complexes. What do we mean protein complexes? Just I discussed before, they are large clusters of protein, and large clusters of proteins are called protein complexes. These protein complexes are photosystems, and we have photosystem 1 and photosystem 2, and we said that they contain the light-absorbing pigments and proteins that play an important role in the light reaction. So that was just everything about photosystems for anyone who wants to, just go over and highlight. Now, next, what do we have? We have the electron transport chain. Electron transport chain. The electron transport chain is a group of proteins in the thylakoid membrane found between photosystem 2 and photosystem 1. I'm going to include a bit of extra information that wasn't necessarily explicitly said in the book. We'll also encounter a couple of things that you might be like, what is that? What is its function? But it is not in our curriculum, or we're not asked to know what these things are, so we don't really need to know their functions. We just need to know what they do for us in this specific reaction. Okay, so we have the electron transport chain. Right, electron transport chain. It is a group of proteins in the thylakoid found between photosystem 1 and photosystem 2. So proteins, a group of proteins found between photosystem 1 and 2. Two and 1. Okay, now we're just going to link to chemistry for a minute. So during the electron transport, electrons will pass from electron donors to electron acceptors to that's it, via the redox reaction. What does this mean? What is all this blabbering that was put here? So during the electron transport, we have electrons that pass from electron donors. What are electron donors? An electron donor is an ion or a molecule that gives away electrons to another ion or molecule. Now, electron acceptors, they do what? It is an ion or a molecule that accepts electrons from another ion or molecule. Now, what is the redox reaction? The redox reaction is reactions where electrons are transferred from one reactant to another. So from one, that basically electrons are transferred from one ion or molecule to the next. Okay? Now, this electron transport is coupled with the transfer of protons, which is the hydrogen plus, and we'll look at that in a minute, from the stroma across the thylakoid membrane into the thylakoid space, and we'll look into that and what that means into a more detailed explanation as soon as we get to the steps of this process. Okay, so we have two more components that we need to go over, and then we can start talking about the steps of this process. So, ferredoxin. We have ferredoxin. Now, ferredoxin is an iron-containing protein which acts as the electron acceptor. And now we know what an electron acceptor is. An electron acceptor associated with photosystem 1. Cool? And then the last thing we have is ATP synthase, and I'm just going to write this here. So we have ATP synthase, and I'm pretty sure you can figure out what it does or its function from the name. We know ATP and we know synthase means to build or to make something, so you've probably concluded that this is an enzyme. So ATP synthase is an enzyme used to synthesize or to make ATP using what we call a proton gradient, and we'll look into more detail into what a proton gradient is in just a minute. Proton gradient. So hopefully now when we're talking about the steps, they will be more clear as we know all of the components and what their functions are. Okay, so let us begin. So the first step here, the first step in the electron transport is you have light energy that excites electrons in photosystem 2. So we start by exciting electrons in photosystem 2. Then the light energy will also cause a water molecule to split. Now you have to keep in mind that there are things that are happening simultaneously, things that are happening at the same time. So the first step is light energy, the light from the sun, will excite electrons in photosystem 2. So we have some electrons in photosystem 2, and what light energy will do is it'll excite them, right? Now, the light energy will also cause a water molecule to split. Now, we know water is H2O. Once the water is split, you will release an electron into the electron transport system. So what you should keep in mind is, when we say the electrons were excited in photosystem 2, imagine photosystem 2 was the house of this electron. If the electrons were kids and the kids got excited, and the kids were inside the house and they got excited and they saw something really fun outside, they're probably going to just run out. So the same thing happens with the electrons. Once the light energy hits them, right, inside photosystem 2, and now they're excited little phonetics in there. Okay, we've got excited electrons inside photosystem 2. They leave. They exit the photosystem. At this point, we've already had a water molecule that is split. And when the water molecule is split, you have a hydrogen ion, which is H+, or in this case also called a proton. Okay. Into the thylakoid space. And when I say into the thylakoid space, that means inside of the thylakoid, not outside it. So you have an H+ or a proton that was released into the thylakoid space, and then the oxygen will go out as a waste product, so it'll leave. Okay, this breakdown of water is essential for photosynthesis to occur. So let's look at what this looks like in the diagram. So right here, this is the first part, which is A, your first step. So the first step, as we said, the light energy is absorbed by photosystem 2. So you can see here, you would have electrons inside photosystem 2. So you've got the electrons inside of photosystem 2. Light energy hits those electrons and it excites them. The light energy will also hit the water molecule inside. And then what will happen to the water molecule? It will break down. And this breakdown of water is essential. It is essential. Once we have a breakdown of water, you're going to have an oxygen molecule be released out, and you'll have 2 H+ or 2 protons released into the thylakoid space, and 1 electron as a resulting of that, or an activated electron. So you'll have an activated electron as a result of that. Now, we said the excited electrons in photosystem 2 will leave, right? They'll leave photosystem 2. Who will take their place? The electron that we got from the water splitting. Now, what happens to the electrons now that they're out of photosystem 2? What happens to them? Well, this got here, and then you have the 2 that have left photosystem 2. What happens to them? Well, the excited electrons are going to move from photosystem 2 to an electron acceptor molecule in the thylakoid membrane, which means this is the membrane, by the way. So you'll have the electrons, and they'll move out of photosystem 2. They'll go to an electron acceptor. Then the electron acceptor will transfer the electrons along a series of electron carriers in photosystem to photosystem 1. So as you can see here, we have these electron carriers right here. And what happens is when your electron leaves photosystem 2, an electron acceptor will take it. It'll be like its taxi, and it will take it through the electron acceptors all the way to photosystem 1. Now what happens? Now what happens to our electrons now that they're at photosystem 1? What goes on? So before that, let me just put this note out here before we forget. So as the excited electrons move down the electron transport chain, so as they go from photosystem 1 through the electron carriers to reach photosystem, so as they go from photosystem 2 to reach photosystem 1, their energy, the energy from these electrons, is used to pump a hydrogen ion from outside, or a hydrogen proton, not an ion, sorry. As the excited electrons move down the electron transport chain from photosystem 2 to photosystem 1, their energy is used to pump hydrogen ions or hydrogen protons from the stroma. So this is the stroma space, okay? This is the stroma out of here, right? And inside here is the thylakoid. So you have hydrogen protons coming from outside of the thylakoid, so from in the stroma space to the thylakoid space. What allows them to go from outside the thylakoid into the thylakoid? The movement of electrons through the electron chain to reach photosystem 1. Now, once they've reached photosystem 1, these electrons have lost most of their energy that they've gained from photosystem 2. So what happens again in photosystem 1 is that light energy will hit these electrons again. Now, the electron is then passed, once it's at photosystem 1, it is then passed to who? It is passed to ferredoxin. And ferredoxin is your final electron acceptor. Okay? Now, the electrons lost by photosystem 1 are replaced by the electrons shuttled from photosystem 2. So, you know, like we lost the electron here, it's left, it's went to ferredoxin. Worry not photosystem 1, because while this is happening at photosystem 1, photosystem 2 is still doing the water splitting and the electron is still moving through the chain. So you can't forget that these processes are all happening at the same time, right? So photosystem 1 has lost an electron, no problem, because photosystem 2, whatever electron has left photosystem 2 will go to photosystem 1 and the process will repeat, right? Okay, so now that we're at ferredoxin, what ferredoxin will do is it'll transfer the electrons to an electron carrier called NADP+. So once you've reached here to ferredoxin, I want to show, where is it? Okay, so at photosystem 1, electrons are rearranged and NADPH is formed. So what happens at ferredoxin? What happens at ferredoxin is we have the NADP+, okay? NADP+ is a compound. Once I add a hydrogen to NADP+, it becomes NADPH, and I have to add an electron, and that will add a proton. So ferredoxin transfers these electrons to a compound called NADPH, okay? And that will form the energy carrier molecule, or the energy storage molecule, NADPH. So NADP+, okay, once you add a hydrogen proton to it and an electron from ferredoxin, because ferredoxin is going to give the electron here, right? Then you're going to have NADP+, plus H plus a proton, that will give you the energy storage NADPH. Okay? Then we have, by now, there is a greater concentration of the hydrogen ions inside, right? We have more hydrogen ions inside of the thylakoid space than outside in the stroma. This creates what we call a proton gradient. And what a proton gradient is, so let's just write that down, proton gradient, so proton gradient. It provides the energy to make ATP in a process called chemiosmosis. So a proton gradient is the uneven distribution of hydrogen ions across two sides of a membrane. So if we see here, this is the thylakoid membrane, right? Outside on this side, this is the stroma. Inside, this is the thylakoid space. In the thylakoid space, I have a lot of hydrogen ions. Outside of the thylakoid space, I don't have many hydrogen ions. So I've created a proton gradient. What does this proton gradient help me in? It helps me in making ATP through a process called chemiosmosis. So what is chemiosmosis? Chemiosmosis is the movement, movement of protons or the hydrogen ions from the thylakoid space to the stroma. And how do they do that? Through what do they pass? They pass through ATP synthase. So this is ATP synthase, and what happens is you have your hydrogen and it'll pass through the ATP synthase. Now, once the hydrogen go down their proton gradient, they go from high concentration to low concentration, so down the proton gradient through ATP synthase. As the protons move through it, their energy is used to convert ADP to ATP. Okay, so that is electron transport completed. As I said, I'll be going over all of the steps once again on the videos that are on ELIF because they're a bit more explanatory, they make a bit more sense, and they're not as confusing as this diagram. But hopefully this allowed for you to understand the diagram a bit better, and it makes the diagram a bit more clear. I'll go over just the steps that are written here so we can just conclude everything, just a last summary of everything that's going on in this chain. So just to summarize and go over all the ideas. So we said step 1 is we have the light energy that is absorbed by photosystem 2 and is used to split a water molecule. And we said the splitting of water is very important. It is essential. So you'd already have electrons in here, and once light hits, these excited electrons will pass through and electron acceptors, which are kind of like their taxi, electron acceptors will take them through the electron carriers. At the same time, a water molecule will split. It'll release an activated electron, it'll release an oxygen, and it'll release 2 hydrogen ions, or 2 hydrogen protons in this case, okay? So this electron that was released by the splitting of water replaces the ones that photosystem 2 has lost. The oxygen is released from the cell, and the protons, which are the hydrogen plus ions, stay in the thylakoid space, and an activated electron enters the electron transport chain. As I said, it will replace the one lost by photosystem 2. Now, the electrons that have left photosystem 2, will be transported to photosystem 1 through the electron carriers, and electron acceptors will be their taxi and take them through. In this process, while the electrons are going through the electron transport chain, this will cause hydrogen ions from outside, from in the stroma, to be pumped inside the thylakoid space. So as the electrons move through the membrane, protons are pumped into the thylakoid space. So now when the electrons have reached photosystem 1, they're drained of all their energy. And so light energy hits photosystem 1, and these electrons are excited again. So these electrons are finally, once they're excited, they're going to leave photosystem 1 and they'll go to the final electron acceptor. And that final electron acceptor is ferredoxin. You should know this. Ferredoxin is the final electron acceptor. Right, what does ferredoxin do? Ferredoxin will transfer the electron to the electron carrier NADP+. So NADP+ is an electron carrier. Cool. And what will happen is, once the electron has left photosystem 1, it's gone through ferredoxin. Ferredoxin will find a NADP+. It will add a hydrogen ion to it and an electron. So ferredoxin will give an electron. To NADP+, and once a hydrogen is added to that, it will turn into NADPH, and NADPH is an energy storage molecule. Okay, energy storage molecule. Right? Then what do we have? When the protons move across the thylakoid membrane through ATP synthase, ADP is converted to ATP. So we said, now that we have more of the hydrogen ions inside the thylakoid, we've created a proton gradient. And we said a proton gradient is when you have a high concentration of protons or hydrogen ions at one side of the thylakoid membrane, and on the other side you have less. So what will happen is, through the process of chemiosmosis, and we said chemiosmosis is movement of protons down their concentration gradient. What does that mean? That means I have more protons inside the thylakoid membrane than I have outside of it, so I'm going down the concentration gradient. And concentration means how much I have. So if I say down the concentration gradient, that means I'm going from more to less. And how do the hydrogen ions leave the thylakoid space? They get pumped through ATP synthase. Now, as the hydrogen ion is going through ATP synthase uses the energy from this process to change ADP. Whoever remembers from the last lesson, we know that ADP needs to be changed into ATP for us to have an energy storage. So ADP will be changed to ATP once the hydrogen is pumped through the ATP synthase. And that is the electron transport chain completed, and that is all that we need to know. Hopefully this has made the lesson a bit easier to understand. You do have to go back and look at it yourself for the ideas to really stick. As I said, I will be putting up another video from Alif, and it'll show you just a bit more in detail and just a better picture, a bit more, less things going on in the picture, and you can really see the animation of how things really happen. So thank you for watching, and I hope this lesson makes sense, and see you next time.